Assalamu alaikum and hello to everybody. Thank you for inviting me into this conference and for giving me the opportunity to talk about my research work from genes to pathophysiology to in utero gene manipulations to precision medicine. Do you know that one of the oldest recognized conditions with written evidence dating back to 4000 before Christ is influenced by genetic alteration? The prevalence of this condition, that is commonly known as epilepsy, to the same extent reflects lack of progress in combating one of the oldest health problems. At cellular level, it results from an abnormal electrical activity within the brain, the most complex and mysterious object in the known universe. Do you know that our brain contains 10 billion tiny computers we call neurons? Each one is connected to 10,000 others through fine wires we call dendrites and axons. Each neuron works like a supercomputer that computes and converts chemical signals into electrical signals using tons of different types of molecular machines we call ion channels. Genetic alterations or abnormal expression of these molecular machines or ion channels are proved to be associated with most of epilepsies. Only 1/3 of epilepsy is due to brain insult, and that is avoidable. However, 2/3 is influenced by genetics and needs genomic medicine. Therefore, understanding of genes and their variants and how they drive health, disease, and drug response. For the last 2 decades, I have been focused on understanding the function of genes expressing ion channels both in healthy and sick children. I also worked on bridging the gaps between mechanistic investigations and complex behaviors by selective manipulations of gene within the uterus using viral genetics, and also bidirectional modulation of different sets of neurons using 2 different approaches. One is called optogenetic and other is called chemogenetic. More importantly, I am establishing personalized medicine that can help combat mysterious conditions associated with many different genetic variants. Currently, I am working on how to rewire brain circuits by delivering genetic information through viruses as scientists developed COVID-19 RNA vaccine. My research journey started in Pakistan with Dr. Peck by looking at hemoglobin variants associated with thalassemia. Then I focused on functional studies of molecular machines or ion channels at Brain Science Institute, KIST, Korea. Here I discovered the role of calcium channel in absence epilepsy. This figure shows functional role of a calcium channel known as CAV 2.3. Previously, researchers classified all calcium channels into 2 groups: low voltage activated and high voltage activated. Based on some properties from non-neuronal studies, this channel was also placed in high voltage activated group. My neuron paper not only settled a long debate of the causal link of calcium channels with genetic epilepsy, but also provided strong evidence that there are 3 classes of channels rather than 2. At Ottawa Hospital Research Institute, Canada, I investigated transcriptional role of LMO4 in calcium ion channels expression. At Schulich School of Medicine, Canada, I investigated role of calcium-dependent potassium ion channel known as PK in sensory filtering related to autism. At Children's Hospital of Philadelphia, one of the finest children's hospitals in the United States, I investigated 28 novel variants of two sodium ion channels related to childhood and infantile epilepsy. Whole exome sequencing led us to the discovery of mutations in SCN3A gene that encodes NAV1.3 and SCN8A gene that encodes NAV1.6, pore-forming subunits of voltage-gated sodium channel. Early infantile epileptic encephalopathy affects newborns within the first few weeks or days of their life. This type of epilepsy is resistant to standard anti-epileptic treatment. Mortality is 50% within weeks to months. Several genes have been associated with infantile epilepsy. It can be associated with mutations in ion channels like KCNCQ2, that is a potassium channel, voltage-gated calcium channels, voltage-gated sodium channels, and likely others. Ion channels are of two types. First is voltage-gated channels and second is ligand-gated channels. Voltage-gated channels are embedded in the cell membranes and open by sensing voltage change that provides a passage through which charged ions can cross the cell membrane. This charges up the whole neuron and the change travels down to axon hillock located at the end of soma that is highly concentrated with sodium channels that fire an action potential or electrical message. Voltage that travels down further to the axon terminals help release neurotransmitters like glutamate or GABA. Second type of ion channels called ligand-gated channels at postsynaptic neurons. Then by sensing released GABA or glutamate neurotransmitters, convert chemical signals into electrical signals. Genetic mutations in these channels can lead to change in their functions, like how fast they should open, how long they should remain open, or how early they get opened, or how long they should stay open. These all can lead to abnormal electrical activity of tiny molecular machine called ion channels. And these ion channels, once altered, they can alter the electrical behavior of neuron. And this could be relayed to one or more circuits of brain. Hyperexcitable circuits of the brain could trigger epileptic seizures that could be detected through EEG and exhibit behavioral changes. Voltage-gated channels are of four types: sodium, potassium, calcium, and chloride. There are several subtypes. Each has unique expression pattern, voltage sensitivity, functional characteristics, and drug sensitivity. Voltage-gated sodium channels play a decisive role in the initiation and propagation of electrical signal in the neurons. In human, there are total 9 sodium channels. 4 sodium channels like 1, 2, 3, and type 6 are predominantly expressed in the brain and are associated with epilepsies. Other 5 are expressed in the spinal cord, peripheral nervous system, and are associated with pain, myotonia, and heart attack. I have engineered and investigated 28 novel variants of channel 3 and 6 that is highly expressed in the brain of epileptic patients, and we collected these variants from all over the world. Mutations in calcium channels are associated with epilepsy, ataxia, autism, and tachycardia. Potassium channels have many different types like voltage sensitive, ATP and calcium sensitive. Potassium channels are key in bringing cells back to rest. I have worked on 2 out of 5 calcium sensitive potassium channels and found that these channels are associated with ataxia and autism. As mentioned earlier, whole exome sequencing led us to the discovery of mutations in the SCN3A and SCN8A genes. Both encode NAV1.3 and NAV1.6, pore-forming subunit of voltage-gated sodium channel type 3 and type 6. For this talk, I will focus on SCN3A work. SCN3A, that is located on chromosome 2, is highly expressed in fetal brain, starting from 16 weeks of gestation. Therefore, in some variants of SCN3A, there are developmental issues reported. SCN3A that starts expressing at week 16 is gradually replaced with SCN1A in postnatal life. Our key finding is to report SCN3A as a new epileptic gene that has not previously been linked to early infantile epilepsy. In this study, we described patients with infantile epilepsy due to de novo mutations in SCN3A gene, that is 116 kb gene, and synthesized 2000 amino acids. So we actually used 6 kb of cDNA, and we introduced all those de novo mutations that we found in epileptic patients. So de novo means something new. That means the variants that we reported appeared as new genetic mutation. This work was mainly conducted at Children's Hospital of Philadelphia. However, there are 25 other institutes that provided mainly whole exome sequencing data. Based on this whole exome sequencing data, we designed a series of innovative work from genetic engineering to channel physiology to in utero gene manipulations to precision medicine. This work has been published in 3 high-profile journals. All 20 de novo point mutations that we studied in SCN3A gene were found in highly conserved region across different species. Here I am showing just one example of the point mutation that led to the substitution of isoleucine to threonine at amino acid level 875. 2 unrelated patients with this mutation had severe epilepsy and polymicrogyria, a condition characterized by abnormal development of the brain before birth. The surface of the brain normally has many ridges or folds. In people with polymicrogyria, the brain develops too many folds, and the folds are usually small, that can profoundly impact cognitive functions and learning. This slide shows an overview of our approaches towards precision medicine. EEG was done to look at epileptic seizure. Whole exome sequencing was performed to look at genetic variations. MRI was done to look at brain abnormalities. Then we engineered SCN3A variants that we found in epileptic patients and expressed in human embryonic kidney cells. Then we characterized these variants electrophysiologically to find out the pathogenic one. Next, we overexpressed pathogenic channels in fetal brain using in utero electroporation and recorded the neuronal firing in the brain slices. In short, combining genomics, physiology, and targeted anti-epileptic drugs, we delivered precision medicine to the patients at Children's Hospital of Philadelphia. To better understand physiology of sodium channel, let's see how it looks like in 2D format. This slide shows structure of pore-forming alpha subunit that contains 4 domains. Each contains 6 transmembrane segments. S4 of each domain is critical in voltage sensing, and the loop between segments 5 and 6 is critical for ion selectivity. And the loop between domain 3 and domain 4 is important in inactivation or closing the channel. So in addition to alpha subunits, there are some beta subunits. They serve as supporting units or auxiliary subunits. And there are 4 different types of beta subunits, like 1, 2, 3, and 4. So the combination of alpha, that is the main subunit that forms the gate, with different other betas also impact its localization in the membrane and other properties. At functional level, sodium channels has 3 conformational states. First is closed, second is active, and third is inactive. In first state, means in closed state, sodium channels are blocked on the extracellular side. In second state, in response to voltage change in the membrane, and that happens mostly due to the ligand-gated ion channels, so the activation gates open. This allows positively charged sodium ions to flow into the cell. In state 3, when there is a peak of action potential, sodium channels inactivate themselves by closing their inactivation gates on intracellular side. Electrical signal on left represents all 3 states, 1, 2, and 3. When channel does not inactivate fully, it is said to be persistently active, leaky, or impaired. In our studies, we found persistent or leaky sodium currents associated with many different variants that we found in epileptic patients. And we predict that this leaky or persistent current could cause increased sodium influx that can cause increased neuronal excitability, and that could lead towards epilepsy. This figure shows just 3 variants out of 20 variants that we studied from epileptic patients. We performed electrophysiological recordings of wild type and compared with the mutants to assess the functional consequences of these variants. The figure A that shows the family of traces showing rapidly inactivating and activating inward currents in response to different test potentials. Just for presentation purpose, there are 3 variants: I875T, V1619A, and P1333L. Figure B shows current density at all different test potentials for few variants. We found 2 abnormalities. One is very clear in the traces, those are shown in the color, and this is impaired inactivation. That means channels are open stuck, causing leak currents into the neurons that in turn could overexcite and cause a surge of electricity in the brain. a characteristic of epilepsy. If we look at IV curves in figure B, peak activation was largely shifted to left compared with the wild type for I875T, that is isoleucine to threonine mutations, and P1333 that caused change from proline to leucine, indicating that these channels could fire an action potential or electrical signal much earlier than the normal one or the wild type. Simply, threshold is messed up. In case of V1769 that caused the valine to alanine change, that shows a huge persistent current and with little early activation. Next, we delivered relatively longer test pulses of 200 milliseconds from holding potential of minus 120 millivolt to test potential at minus 10 millivolt to further clarify the role of NAV 1.3 variations or the variants on sodium persistent current. If you look at the traces, there are striking differences in slowly inactivating or persistent sodium current that is much higher in V1769A and I875T compared with their wild type control. As mentioned earlier, SCN3A expresses in the fetal brain. To assess the impact of epilepsy-associated SCN3A variants on developing brain in the womb, we overexpressed wild type in one case and mutant channel in another case in neocortex by delivering the gene through in utero electroporation, a rapid and powerful approach to generate quick disease model. We performed electrophysiology in the brain that received wild type in one case and mutant genes in another case while they were sitting in the uterus. Then we assessed intrinsic firing properties of neocortical neurons overexpressing wild type versus mutant SCN3A. If we look at the traces, the current clamp recordings revealed spontaneous bursting from resting membrane potential in the cells that are expressing mutant channel, but not in the cells overexpressing wild type channel. Such results further validate the pathogenic nature of the epilepsy-associated SCN3A variant. Targeting persistent current is a proposed therapeutic strategy in epilepsy. As mutant channels showed increased persistent current relative to wild type, we hypothesized that pharmacological agents that target persistent sodium current might normalize epilepsy-associated sodium currents and could represent potential treatments for SCN3A encephalopathy. We tested phenytoin, commonly known as dilantin, lacosamide, that is newly synthesized and approved drug, and carbamazepine, that is commonly used. We found that lacosamide blocked specifically pathogenic currents more in I875T than V1769A patient. On the other hand, phenytoin or dilantin produced a significant decrease in pathogenic sodium persistent current of the mutations V1769A. Based on these findings, we delivered precision medicine to CHOP epileptic patients by knowing their genetics. Strangely, in one case, all anti-epileptic drugs failed and the parents refused to try any new drug. However, we found combination of drugs in the laboratory setting that could address the issue. And then we convinced the parents, if we try this combination, this could help reduce a pathogenic sodium current that we have not seen in any other variants. And this was actually the much bigger sodium influx. And there was much bigger left shift. So when we tried the combination, it worked. Based on these findings, we proposed and received another grant of $2 million from NIH USA to further look at the role of healthy and pathogenic variants of SCN3A in brain development. Towards this, we already have generated CRISPR-mediated rodent disease models. For patients, the Children's Hospital of Philadelphia already has started engine that starts for Epilepsy Neurogenetics Initiative. Neurons derived from patients' induced pluripotent stem cells are being tested for precision medicine, an approach to select treatments that are most likely to help patients based on a genetic understanding of their disease. I think soon we will be allowed to edit the genome through CRISPR to prevent severe forms of genetic disorders. I can imagine strong criticism for those who have healthy genetic makeup, and can imagine excitement in those who are suffering from greater pain.